NCBI-GEO, a gene expression database. Here, we will be looking at how to access, download, and modify DNA microarray data in preparation for analysis from the NCBI-GEO database. Specifically, the data used will be research on the gene expression of cancerous thyroid cells with the accession number GSE29265. This data was obtained from the cited research paper on papillary thyroid cancer, Amjad et al., 2023. In order to access and prepare the data for analysis, I will show you how to download data from the NCBI database and explain the code needed to make the data accessible and readable, as well as preparing it for analysis. Why is NCBI-GEO important? Gene expression data is important because it provides insight into how cells differentiate, interact with their surroundings, and develop. The specific pattern of genes turned off or on in a cell determines the cell's function and identity, muscle cell, skin cell, etc. Because of this, comparing the gene expression of healthy cells to that of diseased cells can show which alterations in gene expression cause problems, paving the way for future medical interventions. This makes NCBI-GEO important because it is one of the main databases for gene expression data, specifically RNA sequencing data, also called RNA-seq, and DNA microarray data. Acquiring the data. Acquiring the data from NCBI-GEO has two steps, which will be shown in picture form in the next two slides. Step 1. First off, we have to go to the NCBI homepage, find the GEO datasets in the All Datasets tab, and type the accession number into the search bar. Then we click on the first link to find the dataset we need. Step 2. Next, we need to scroll down to the bottom of the dataset page and click on the link in the second picture with the cursor hovering over it to download the data. Here is the picture of the main home page from which we go to the GEO dataset and type in the accession number. And here is where we download the link, right here. Now we're talking about accessing the data. When the data is downloaded from NCBI-GEO, it's in tar format. So, in order to make the data accessible, we need to untar it into a number of .cell files, which then need to be turned into a readable data frame. The R code needed to do this is shown in the highlighted section on the next slide, and goes through getting, untarring, and reading in the data as shown in the comments. Here is the code with the comments telling you what it's doing. Normalizing the data. Now that we've gotten the data and made it readable, the next step is to prepare the data for analysis and make it usable. The first step in that process is to normalize the data by gene, so as to make differences in expression clear and easy to understand. The R code for this is shown in the highlighted section in the picture on the next slide. And here is the code for normalizing the data. Okay, now we're going on to mapping the data. So, the data has been normalized. Great. Now we need to convert the data to a data frame and take the two features of the data that we care about, probe ID and gene symbol, and map them to each other. Now our new dataset is set up with our two important features already prepared to be separated out. The code for this can be seen in the highlighted section on the next slide. Here is the code. Okay, the two tables, feature data and normalized expression. Now that we've gotten the data frame set up with the probe IDs and gene symbols mapped to each other, it's time to focus on making the two tables we need to analyze the data. Table 1. The first table is called feature data. This table is a subset of the data frame we just made, containing only the two features, probe ID and gene symbols, that we care about. The code for creating this is shown on the next slide in the highlighted section. Table 2. The second table is called normalized expression. This table is not a subset of the data frame, but is instead made from the normalized data. Here, the row names and columns are flipped, and the table shows the gene expression by showing the data normalized by gene. The code for creating this is shown on the slide after next in the highlighted section. Here is the first slide for feature data, and here's the second slide for normalized expression. the final tables. Congratulations! We've managed to acquire, access, and prepare the data for analysis. You now have two tables. Table 1, data relating to the features you need, probe ID and gene symbols, and Table 2, the gene expression data for all the genes. This means that now we have the data we need to tell us all about the gene expression of cancer thyroid cells. After everything, the first few rows for each of the two tables you've made should be similar to the one shown in the images on the following two slides. Here is an image of the first few rows of the feature data table, and here is an image of the first few rows of the normalized expression data table. So, conclusion. We've now learned how to use the NCBI-GEO database and how to provide and how to process and analyze the datasets you acquire from it. This should show you how useful the NCBI-GEO database can be in studying gene expression and in bioinformatics research in general. And here are the citations. The end.